Continuation of the previous video, like remote sensing methods. Satellite imagery. Satellite imagery requires equipment such as satellites and sensors such as cameras and spectrometers. The procedure involves collecting data on the Earth's surface from space. Applications include mapping data, land use and land cover, monitoring vegetation health and productivity, and assessing the impacts of natural disasters such as wildfires and floods. Satellite imagery is important for monitoring large-scale environmental changes and for informing land management decisions. Health, productivity, and assessing the impacts of natural disasters such as wildfires and floods. Satellite imagery is important for monitoring vegetation health and productivity and assessing the impacts of natural disasters such as wildfires and floods. Satellite imagery is important for monitoring large-scale environmental changes and for informing land management decisions. So LIDAR requires equipment such as a laser scanner mounted on an aircraft or drone. The procedure involves emitting laser pulses and measuring the time it takes for them to bounce back, which provide information on the elevation and structure of the Earth's surface. Applications include mapping topography, monitoring forest biomass and carbon storage, and assessing the impacts of natural disasters such as landslides and earthquakes. LIDAR is important for producing high-resolution topographic maps and for informating the land and management decisions. Like radar. Radar requires equipment such as a radar antenna mounted on an aircraft or satellite. The procedure involves emitting radio waves and resulting the time it takes for them to bounce back, which provides information on the Earth's surface features. Applications include monitoring ocean currents, ice cover, changes in land surface due to earthquakes or volcanic eruptions. So what is the use of radar? Radar requires equipment such as a radar antenna mounted on an aircraft or satellite. The procedure involves emitting radio waves and measuring the time it takes for them to bounce back, while provides information about its surface features. Applications include monitoring ocean currents, ice cover, and changes in land surface due to earthquakes or volcanic eruptions. Radar is important for monitoring large-scale environmental changes and for informing management decisions related to climate change and natural hazards. The importance of both ground and remote sensing methods lies in their ability to provide large-scale, accurate, and timely data on environmental parameters. This data is critical for environmental monitoring and management, such as identifying areas of environmental concern, tracking changes over time, and evaluating the effectiveness of management strategies. By combining ground and remote sensing methods, we can gain a more comprehensive understanding of the Earth's environment systems and make more informed decisions about their management. Ground and remote sensing methods are essential techniques for studying various ecological phenomena. This method requires specialized equipment and procedures to be effective and may have numerous applications and benefits. Ground sensing methods require making measurements and observations directly on the ground. These methods may include transect sampling. The transect is a straight line that is used to sample an area. Transect sampling involves counting or observing organisms or features along a transect line. This method is commonly used for estimating population densities to study changes in vegetation over time. Quadrant sampling. First one is the transect sampling. Just remember a line. Transect sampling is there. Transect sampling. A transect is a straight line that is used to sample an area. Transect sampling involves counting or observing organisms or features along a transect line. This method is commonly used to estimate population densities or to study changes in vegetation cover over time. Next, quadrant sampling. A quadrant is a small square or rectangle method is used to sample an area. Quadrant sampling involves counting or observing organisms or features within the quadrant. This method is commonly used to estimate plant populations or to study changes in soil composition. Next, mark recapture method. This method involves capturing and making a sample of organisms, releasing them back into the environment, and then capturing a second sample at a later stage. By comparing the number of marked individuals in the second sample to the total number of individuals in the sample, scientists can estimate the population size. Next, remote sensing methods involve using technology to observe and measure features from a distance. Next is the mark recapture method. This method involves capturing and marking a sample of organisms, releasing them back into the environment, and then recapturing a second sample at a later time by comparing the number of marked individuals in the second sample to the total number of individuals in the sample. Scientists can estimate the population size. Remote sensing methods involve using technology to observe and measure features from a distance. Satellite imagery with LIDAR, like light detection and ranging. Technology uses laser to measure distances and create detailed 3D maps of the Earth's surface. This involves commonly used to study changes in elevation or vegetation cover. Thermal imaging. Thermal imaging cameras can detect differences in temperature, which can be used to study the behavior of animals or to detect changes in vegetation or water quality. Both ground and remote sensing methods have numerous applications in ecological research, including monitoring changes in ecosystems. Ground and remote sensing methods can be used to monitor changes in ecosystems over time, including changes in vegetation cover, water quality, or animal populations. Next, mapping and modeling. Ground and remote sensing methods can be used to create detailed maps and models of ecosystems, allowing scientists to better understand the complex interactions between organisms and their environment. Conservation and management. Ground and remote sensing methods can be used to inform conservation and management efforts, including identifying areas of high biodiversity or monitoring the effectiveness of conservation programs. In conclusion, ground and remote sensing methods are critical tools for ecological research. These methods include specialized equipment and procedures, but they offer numerous benefits, including the ability to monitor changes in ecosystems, create detailed maps and models, and inform observation and management efforts. Next, population growth curves in detail. Population growth curves are mathematical representations of the changes in population size over time. These curves are essential for understanding how populations change over time and how factors such as resource availability, predation, and disease can impact population growth. There are two main types of population growth curves. First one is the exponential growth curve, and next is the logistic growth curve. So these growth curves are mathematical representations which represent the changes in population size over time. We are concentrating on the population size, change of population size over time. These curves are essential for understanding how populations change over time and how factors such as resource availability, predation, and disease can impact population growth. So first, exponential growth curve occurs when a population is growing at an accelerating rate, which with each generation producing more offspring than the previous generation. The curve for exponential growth is typically a J-shaped curve indicating rapid population growth. Examples of population exhibit exponential growth include bacteria and other microorganisms, as well as some invasive species in new environments. While exponential growth can be beneficial for a population in short term, it is not sustainable in a long term due to resource limitations. Next is the logistic growth curve. Logistic growth curve occurs when a population grows rapidly at first, but then reaches a carrying capacity, a maximum population size that the environment can support. The curve for logistic growth is typically an S-shaped curve, indicating that population growth initially accelerates, then levels off as carrying capacity is reached. So, examples of populations that exhibit logistic growth curve include many plant and animal populations. Understanding the carrying capacity of an environment is essential for managing wildlife populations and other natural resources. The importance of population growth curves lies in their ability to help scientists and resource managers understand the dynamics of populations over time. By studying these curves, researchers can identify patterns in population growth and fluctuations and make predictions about future population changes. Population growth curves are also playing a critical role in the management of wildlife populations and their conservation for endangered species. For example, consider the population growth curve of a gray wolf population in Yellowstone National Park. When wolves were reintroduced into the park in the 1990s, the population initially exhibited exponential growth. However, as the wolf population approached carrying capacity, growth slowed and eventually leveled off. By studying the population growth curve, wildlife managers were able to understand the carrying capacity of the park for wolves and develop management strategies to maintain a sustainable population. In conclusion, population growth curves are important tools for understanding population dynamics, resource management, and conservation efforts. By studying these curves, researchers can gain insight into how populations change over time and how environmental factors impact growth rates. Population growth curves are critical for the sustainable management of natural resources and the conservation of endangered species. Next, life tables. Life tables are an important tool for studying population dynamics and the life history of a species. They are statistical models that provide information on the survival and mortality rates of individuals within a population, as well as other key life history parameters such as fecundity and age at first reproduction. Life tables are typically constructed for a single cohort of individuals with data collected over a specific time period. There are two types of life tables, static and dynamic. Static life tables are constructed using data from a single snapshot in time, while dynamic life tables are constructed using data collected over time and account for changes in population structure. So, dynamic life tables are constructed using data collected over time and account for changes in population structure. Life tables are important because they provide a wealth of information about the life history of a species, including the age structure of the population, the rates of mortality and survival, and the reproductive output of individuals. By analyzing this information, researchers can gain insights into factors that affect population growth and survival, such as predation, disease, and resource availability. Here are a few examples of the importance of life tables. First is the conservation. Life tables can be used to assess the viability of endangered populations and to develop management strategies to ensure their survival. For example, life tables were used to assess the viability of the Florida panther population and to develop a captive breeding program to increase genetic diversity and improve survival rates. Evolutionary biology. Life tables can provide insight into the evolution of the life histories and reproductive strategies. For example, studies of life tables have shown that some species have evolved to produce large number of offspring with low survivorship rates, while others produce fewer offspring with higher survivorship rates. Human population studies. Life tables are also used to study human populations and to develop public health policies. For example, life tables are used to estimate life expectancy and mortality rates, which can be used to develop healthcare policies and to track the impact of diseases and other health-related issues. In conclusion, life tables are an important tool for studying population dynamics and the life history of a species. They provide valuable insights into factors that affect population growth and survival, as are used in a variety of fields including conservation biology, evolutionary biology, and public health. By analyzing data from life tables, researchers can develop management strategies to ensure the survival of endangered populations, study the evolution of the histories, and develop policies to improve human health and well-being. Next, life tables. There are two types of life tables, static and dynamic. So static life tables. Static life tables are constructed using data from a single snapshot of time and are used to estimate the survival and mortality rates of population over a specific time period. They provide a snapshot of the population structure at a particular point in time and are often used to compare different populations or to study differences in mortality and survival rates across different age groups. Static life tables are constructed by recording the number of individuals at each age class in the population and the number of individuals and die in each age class during a specific time period. So static life tables are constructed by recording the number of individuals at each age class in the population and the number of individuals that die in each age class during a specific time period. The proportion of individuals that survive from one age class to the next is then calculated along with the mortality rate for each age class. Next, dynamic life tables. Next is the dynamic life tables. Dynamic life tables are constructed using data collected over time and account for changes in population structure. They provide more accurate estimate of survival and mortality rates and are often used to study population dynamics and to develop conservation strategies. Dynamic life tables are constructed by following a cohort of individuals from birth to death, recording the number of individuals at each age class and the number of individuals that die during each time interval. The survival rate is calculated by dividing the number of individuals that survive from one time interval to the next by the total number of individuals in the previous time interval. Dynamic life tables are more complex to construct than static life tables, but provide more accurate estimates of the population dynamics and are better suited for studying long-term changes in population structure and mortality rates. In summary, static life tables provide a snapshot of the population structure at a particular point in time, while dynamic life tables account for changes in population structure over time. Both types of life tables are important tools for studying population dynamics and the life history of a species and are used in a variety of fields, including conservation biology, evolutionary biology, and public health. Next, survivorship curves. Survivorship curves are graphical representations of number of individuals surviving at each class in a population. They are important tools for understanding the life history of a species and for predicting the future population growth and stability. There are three types of survivorship curves: type 1, type 2, and type 3. Type 1 survivorship curve. Type 1 survivorship curve are characterized by high survival rates in the early and middle life stages with a steep decline in survival rates in the later stages. This type of survivorship curve is typically for large mammals including humans, elephants, and whales. The species typically have long lifespans, low reproductive rates, and high parental investment in offspring. And maybe that parental investment is a factor which is responsible for the early survival rate. So type 1 survivorship curves are characterized by high survival rates in the early and middle life stages with a steep decline in survival rates in the later stage, for example humans, elephants, and whales. Type 2 survivorship curve. Type 2 survivorship curve are characterized by relatively constant mortality rates throughout the lifespan. This type of survivorship curve is typically of birds, small mammals, and reptiles. The species typically have moderate lifespans, moderate reproductive rates, and moderate parental investment in offspring. Type 3 survivorship curve. Type 3 survivorship curve are characterized by low survival rates in the early life stages, followed by a period of high survival rates in the middle stages. Type 3 survivorship curve. Type 3 survivorship curve are characterized by low survival rates in the early life stages, followed by a period of high survival rates in the middle stages and a decline in survival rates in the later stages. This type of survivorship curve is typical of insects, plants, and fish. This species typically have short lifespans, high reproductive rates, and low parental investment in offspring. The importance of survivorship curves lies in their ability to predict the future population growth and stability of a species. By examining the shape of the curve, it is possible to determine the age structure of the population and the potential for population growth or decline. For example, a population with a type 1 survivorship curve is predicted to have a stable or slowly growing population, while a population with a type 3 survivorship curve is expected to have a rapidly growing population, but and that is susceptible to environmental fluctuations. Examples of survivorship curves include type 1 humans, elephants, and whales. Type 1 example human, elephant, whale. Type 2 birds, rodents, and reptiles. Type 3 insects, plants, fish. In summary, survivorship curves provide important information about the life history of a species and its potential for population growth and stability. Understanding survivorship curve is essential for conservation efforts and the management of wild populations, as it allows for the prediction of population dynamics and the identification of key factors affecting population growth and survival. Population regulation types in detail. Population regulation refers to the mechanisms that control the growth and size of populations. These mechanisms can be intrinsic or extrinsic and can act on various levels of the population. Here are the types of population regulation. Density dependent regulation. Density dependent regulation occurs when population growth rates are affected by a density of individuals within a population. As population density increases, resources become more scarce, competition for resource increases, and stress-related factors like predation, disease, and parasitism become more prevalent. This results in a decrease in the growth rate of population. Some examples of density-dependent regulation include competition for food among animals in a population, such as deer competing for access to limited food resources during the winter. Disease outbreaks in populations, such as when a virus spreads rapidly among a group of individuals living in a close proximity. Predator-prey dynamics. Where an increase in predator populations can lead to a decrease in prey populations and vice versa. Density-dependent regulation. Density-dependent regulation occurs when population growth rates are affected by external factors that are not related to the density of the population. These factors can include environmental conditions such as weather patterns, natural disasters, and habitat destruction. Density-independent factors affect populations in a similar way, regardless of their density. Some examples of density-dependent regulation include droughts that lead to decreased water availability and reduced plant growth, which can have a cascading effect on other populations that depend on these plants for food and shelter. Forest fires that destroy large areas of habitat and disrupt ecosystems, leading to declines in populations of animals and plants. Climate change, which can alter environmental conditions and disrupt the balance of ecosystems, leading to changes in population size and distribution. Human regulation. Human regulation refers to the ways in which human activities can impact populations of other species. This can include habitat destruction, pollution, overhunting. This can include habitat destruction, pollution, overhunting or overfishing, and introduction of non-native species. Human regulation can have a significant impact. This can include habitat destruction, pollution, overhunting or overfishing, and introduction of non-native species. Human regulation can have a significant impact on populations and can lead to declines in species, loss of biodiversity, and ecosystem disruption. Examples of human regulation include habitat destruction caused by deforestation, urbanization, and agricultural activities. Next, pollution of air, water, and soil that can lead to ecosystem disruption and harm populations of plants and animals. Overhunting or overfishing of species that can lead to declines in population size and even extinction. Next, introduction of non-native species that can outcompete native species and disrupt the balance of ecosystems. The importance of understanding population regulation lies in its role in maintaining biodiversity and ecosystem functioning. By understanding the mechanisms that regulate population growth and size, conservation efforts can be directed towards maintaining healthy ecosystems and preventing species loss. In addition, understanding population regulation is important for managing populations of species that are of economic or ecological importance, such as game animals or pollinators. Then let's have a quick recap about what we just studied. That is population regulation types. It can be of intrinsic or extrinsic type. First, density dependent regulation. Then predation, disease and parasitism become more prevalent. Competition for food. Then disease outbreaks. Predator prey dynamics. Density independent regulation. These three are density dependent regulation. Density dependent regulation examples are competition for food among animals in a population, disease outbreaks in populations, predator prey dynamics. Next, density independent regulation, which are related to environmental conditions like droughts, forest fires, and human regulation or climate change along with human regulation. And deforestation, urbanization and agricultural activities, pollution of air, water and soil, overhunting or overfishing of species. And these are the examples. Next, life history strategies types explanation in detail. Life history strategies refers to the adaptive traits. Life history strategies refers to the adaptive traits and trade-offs of an organism which are determined by a combination of genetic and environmental factors. These traits are related to the reproductive characteristics of the organism and their lifespan. Here are the types of life history strategies. Are selected species. These species have a high reproductive rate and short lifespan and small body size. They tend to produce large number of offspring but provide little parental care. Examples of R-selected examples include insects, weeds and some fish. K-selected species. These species have a lower reproductive rate, a longer lifespan, lower reproductive rate, a longer lifespan and a larger body size. Lower reproductive rate, they have a lower reproductive rate, a longer lifespan will be there and a larger body size. They produce fewer offspring but invest more in parental care. Examples of K-selected species include humans, elephants and whales. So which are the K-selected species? K-selected species have a lower reproductive rate, a longer lifespan and a larger body size. K-selected species. These species have a lower reproductive rate, a longer lifespan and a larger body size. They produce fewer offspring but invest more in parental care. Examples of K-selected species include humans, elephants and whales. Semel-pair species. These species reproduce once in their lifetime, typically with a large number of offspring and then die. Examples include some insects, salmon and some plants. Next, heteropair species. These species reproduce multiple times throughout their lifetime with smaller numbers of offspring. They tend to have longer lifespans and invest more in parental care. Examples include humans, elephants and many birds. Which are the semel-pair species? Produce only once in their lifetime, typically with a large number of offspring and then they die. For example, insects, salmon and some species. Next, heteropair species. Heteropair species. These species reproduce multiple times throughout their lifetime with smaller number of offspring. They tend to have longer lifespans and invest more in parental care. Examples include humans, elephants and many birds. The importance of understanding life history strategies is that they provide insights into the adaptive traits and trade-offs that organisms make in response to the environmental pressures. These strategies influence the survival and reproductive success of a species, and understanding them can help in the conservation and management of populations. Once again, what is heteroparous species? These species reproduce multiple times throughout their lifetime with smaller number of offspring. They tend to have longer lifespans and invest more time in parental care. Examples: humans, elephants, and many birds. The importance of understanding life history strategies is that they provide insight into the adaptive traits and trade-offs that organisms make in response to environmental pressures. These strategies influence the survival and reproductive success of a species, and understanding them can help in the conservation and management of populations. For example, understanding the life history strategies of a particular species can help in predicting how it will respond to habitat destruction or other environmental changes. Understanding the life history strategies of a particular species can help in predicting how it will respond to habitat destruction or other environmental changes. R and K selection in detail with multiple examples and importance. R and K selection are two contrasting reproductive strategies that are exhibited by different species in response to different environmental conditions. These strategies were originally proposed by the ecologists Robert MacArthur and E.O. Wilson in the early 1960s. R-selected species are those that prioritize high reproductive rates and rapid growth over survival in the face of environmental challenges. They tend to have a shorter lifespan, mature early, and reproduce a large number of offspring and little or no parental care. They thrive in unstable and unpredictable environments with high levels of disturbance, such as rapidly changing weather patterns, fire, or floods. Examples of R-selected species include insects, rodents, some plants, and most marine organisms. Then K-selected species. K-selected species, on the other hand, prioritize survival and growth over rapid reproduction. They tend to have a longer lifespan, mature later, and reproduce fewer offspring with higher level of parental care. They thrive in stable environments with fewer disturbances, where competition for resources is high. Examples of K-selection species include large mammals like elephants, primates, and some birds. The importance of understanding R and K selection lies in their application to ecological management and conservation. For example, in conservation biology, understanding R and K selection can help in predicting the effects of environmental changes on populations of different species. It can also help in designing management plans that are appropriate for the different reproductive strategies. For example, R-selected species may require more frequent habitat restoration efforts to recover from disturbances, while K-selected species may require longer periods of undisturbed habitat and thrive. In conclusion, R and K selection are two contrasting reproductive strategies that are exhibited by different species in response to different environmental conditions. Understanding these strategies can provide insights into how different species respond to environmental changes, which can help in the management and conservation of populations. So in conclusion, R and K selection are two contrasting reproductive strategies that are exhibited by different species in response to different environmental conditions. Understanding these strategies can provide insight into how different species respond to environmental changes, which can help in the management and conservation of populations. Demes and dispersal types in detail. Demes and dispersal are important concepts in population ecology that describe the movement and distribution of individuals within a population. Demes. A deme is a small local population of interbreeding individuals that are connected by gene flow. Deme is a small local population of interbreeding individuals. They interbreed. Demes and dispersal are important concepts in population ecology that describe the movement and distribution of individuals within a population. So, a deme is a small local population of interbreeding individuals that are connected by gene flow. Demes can be considered as the building blocks of larger populations and are often used to study genetic diversity and the effects of local environmental conditions on population dynamics. Demes can be classified into two types: closed demes Closed teams are isolated populations that do not exchange genes with other populations. Closed teams are isolated populations that do not exchange genes with other populations. They are often found in islands or in isolated habitats such as mountain tops or deserts. Examples of closed teams include the Galapagos finches and the lemurs of Madagascar. Open teams. Open teams are populations that exchange genes with other populations. Open teams just exchange their genes with the outer population. For example, in case of closed teams, we have learned the example of Galapagos finches and the lemurs of Madagascar because they are isolated from the entire population, entire other population, so their genes just remain in that, but they do interbreed in between them. So open teams are populations that exchange genes with other populations. Those populations are connected by gene flow, which helps to maintain genetic diversity and prevent inbreeding. Examples of open teams include populations of migratory birds, fish, and mammals. These populations are connected by gene flow, which helps to maintain genetic diversity and prevent inbreeding. Examples of open teams include populations of migratory birds, fish, and mammals. Migration, they undergo migration, and by that process they just disperse their genes wider, and this will just decrease the inbreeding. So dispersal refers to the movement of individuals from one location to another. Dispersal is an important ecological process that helps to maintain genetic diversity, colonize new habitats, and avoid competition. Dispersal can be classified into two types, like active dispersal is there. Active dispersal occurs when individuals move under their own power, such as flying, swimming, or walking. Active dispersal occurs when individuals move under their own power, such as flying, swimming, or walking. Examples of animals that exhibit active dispersal include birds, fish, and mammals. Passive dispersal occurs when individuals are transported by external factors such as wind, water, or other organisms. Examples of animals that exhibit passive dispersion include seeds carried by the wind, larvae carried by ocean currents, and animals that attach in the fur of other animals. The importance of understanding teams and dispersal lies in their application to conservation biology. So what are teams? They are just interbreeding populations which are connected with genes. There are two types, like closed teams, which are which located in the islands or some regions like that, and lemurs of Madagascar and Galapagos Island finches. They just don't they are not capable of distributing their genes outside of that zone, so their genes are just remain there. There will be a greater amount of inbreeding. So that's about closed teams. But open teams, they just exchange their genetic material with other organisms, like with the help of migration on all that. So migratory birds are example, migratory animals, and some fishes are example of open teams. Dispersal is of two types, like active and passive. Active in the sense they are voluntarily moving under their own power, flying, swimming, or walking. They just voluntarily doing that migration process, dispersal process. But on the other hand, when we talk about the passive dispersal, this is not happening, as this passive dispersal is happening with the help of some external agents like wind, water current, or whatever it may be, like attaching in the surface of animals, etc. So the importance of understanding teams and dispersal lies in their application to conservation biology and habitat management. By studying the genetic structure and movement patterns of populations, ecologists can determine which areas are important for maintaining genetic diversity and which areas are critical for the survival of endangered species. This information can then be used to design conservation strategies that protect and restore these areas. In conclusion, teams and dispersal are important concepts in population ecology that describe the movement and distribution of individuals within a population. Understanding these concepts can help to explain patterns of genetic diversity and inform conservation and habitat strategies. Next, interdemic extinctions. Multiple examples. Interdemic extinction is a type of extinction event that occurs when multiple populations within a species go extinct in a fragmented habitat, leading to the loss of the species as a whole. This process is often driven by habitat fragmentation, which creates isolated populations that are more vulnerable to local environmental changes such as disease outbreaks or natural disasters. The importance of interdemic extinction lies in their potential to contribute to a broader phenomenon known as the sixth mass extinction, which refers to the ongoing extinction crisis currently underway on Earth. Interdemic extinction can have cascading effects on entire ecosystems, leading to the loss of critical environmental services and disrupting food webs and nutrient cycles. Multiple examples of interdemic extinction exist throughout history. One of the most well-known examples is the extinction of the woolly mammoth, that is Mammuthus primigenus, at the end of the East Ice Age. As the climate warmed and the ice sheets retreated, the mammoth's habitat became increasingly fragmented and the species was unable to adapt quickly enough to survive. Similarly, the extinction of the dodo, that's Rhapis cucullatus, in the 17th century was driven in part by the fragmentation of its habitat on the islands of Mauritius, which left the population vulnerable to hunting and invasive species. Interdemic extinctions are not limited to terrestrial species. They can also occur in aquatic environments. For example, the Yangtze River dolphin, Lipotus vexillifer, is believed to have gone extinct due to interdemic extinctions caused by habitat fragmentation and pollution. The fragmentation of the dolphin's habitat into isolated populations combined with human activity that polluted the river made it difficult for the species to survive. Overall, interdemic extinctions are an important component of an ongoing mass extinction crisis. The understanding their causes and effects is critical for the conservation of endangered species and the maintenance of biodiversity. Growth and regulation of human population in detail with advantages, disadvantages, and control measures and importance. The growth and regulation of the human population has been a topic of concern for many years due to its importance in the impacts on the environment, ecology, and society. In this essay, we will discuss the growth and regulation of the human population, including its advantages, disadvantages, control measures, and importance. The human population has been increasing rapidly since the 80th century due to factors such as advancements in medical technology, improvements in sanitation, and increased food production. The current global population is over 7.8 billion people and is projected to reach 9.7 billion by 2050. The growth of the human population has many advantages such as increased economic growth, increased diversity, and increased cultural exchange. One of the disadvantages of the growth of human population is increased pressure on natural resources such as water, food, and land. This has led to environmental degradation, deforestation, and loss of biodiversity. Additionally, the growth of the human population has led to increased pollution, which has negative impacts on the environment and human health. Furthermore, overpopulation in some regions has led to social and economic problems such as poverty, unemployment, and overcrowding. There are various control measures that have implemented to regulate the growth of the human population. One of the most effective methods is education, which can help to increase awareness about family planning and the benefits of having smaller families. Family planning programs, including the provision of contraceptives and other reproductive health services, have also been successful in controlling population growth. Other measures such as immigration policies, economic incentives, and social programs have also been used to control population growth. The importance of regulating the growth of the human population cannot be overstated. Regulating population growth can help to ensure that natural resources are used sustainably and that there is enough food, water, and land to meet the needs of future generations. Additionally, regulating population growth can help to reduce social and economic problems such as poverty, unemployment, and overcrowding. In conclusion, the growth and regulation of the human population is a complex issue with both advantages and disadvantages. Control measures such as education, family planning programs, and immigration policies have been successful in regulating population growth. The importance of regulating population growth cannot be overstated as it helps us to ensure that natural resources are usually sustainably used and social and economical problems are reduced. So that's it about this chapter about population ecology. Thank you for listening.